I'll get started here. So welcome and thank you for joining us this evening for today's webinar, April 2nd, 2025. I'm Marcia Baker, the Program Director for the SADS Foundation. This webinar is being sponsored by Tenaya Therapeutics. The SADS team has been partnering with Tenaya Therapeutics patient advocacy team for several years now as investigational therapies for arrhythmogenic cardiomyopathy are being studied. Dr. Whit Tingley will provide us an overview of Tenaya's clinical trials for HCM and ARVC, and you'll have a chance to ask questions at the end of the presentation. First, let me introduce Dr. Tingley. He joined Tenaya as the Chief Medical Officer in December of 2018. Previously, he served with Cytokinetics as Vice President of Clinical Development, Cardiology, and at Genentech as Therapeutic Area Lead for Cardiovascular and Metabolism Research and Early Development. Whit formerly was Adjunct Associate Professor of Medicine in the Cardiology Division at the University of California, San Francisco, and cared for patients as an attending cardiologist in the UCSF Cardiology Faculty Practice. Dr. Tingley earned his Bachelor of Arts degree from Brown University and his MD and PhD degrees from Johns Hopkins University School of Medicine. He completed internship and residency programs at the Johns Hopkins Hospital, a cardiology fellowship at UCSF, and postdoctoral research at the Gladstone Institutes. We are honored that Whit is joining us tonight. We thank you, our community, for participating as well and hearing firsthand from Tenaya. Following Whit's presentation, you'll have plenty of time to ask questions by posting them in the chat feature located on the bottom of your Zoom screen. A reminder that this webinar is being recorded. Now, Whit, I'll turn this presentation over to you.
Thank you, Marcia and Genevieve, everyone at SADS for inviting us to present our interim data coming out of our gene therapy trials. Really very much appreciated, and certainly appreciate the partnership and everything that you do for patients affected by arrhythmias. And yeah, thanks to everyone who's joined today. Thank you for your interest in what we're learning about gene therapy for cardiomyopathies. So the agenda will go through relatively quickly here to make sure we leave time for your questions at the end. Please do save up your questions. I really look forward to that part of the meeting. We'll talk briefly about ourselves at Tenaya Therapeutics, give some primer on how gene therapy works using a delivery method called AAV. We'll talk about results for our lead gene therapy program, the one first into clinic, which is addressing a genetic form of hypertrophic cardiomyopathy, or HCM. Then we'll talk about the implications of that for our second gene therapy, which is for arrhythmogenic right ventricular cardiomyopathy, in particular patients affected by mutations in the PKP2 gene. So at Tenaya, our mission is to transform and extend lives through discovery, development, and delivery of potentially curative therapies that target the underlying cause of heart disease. We are 100% focused on heart disease. We have 5 clinical trials ongoing, including 2 active gene therapy studies. And we very much value our partnership with patients through the process of developing drugs and partnering directly with patients. We have our patient advocacy team and a patient advisory council to give us guidance, and we also partner with foundations such as SADS. There are many steps along the way in drug and biologic development that patients can help with the process, and if you are interested in participating in any way, please do let us know. And you can contact Wendy Borsari, our head of patient advocacy and also affected by genetic cardiomyopathy, by contacting her at patient.advocacy@tenayathera.com. patient.advocacy@tenayathera.com. So gene therapy is an investigational approach to potentially treat genetic heart conditions. The goal is to deliver a new working copy of a gene when there is a copy in the heart that isn't functioning properly. And we can see in our preclinical animal models that this can reverse adverse effects of genetic mutations, can improve heart function even after onset of disease, as well as prevent the development of cardiomyopathy and arrhythmias. By doing this, we're targeting the underlying cause of disease. For both groups of patients we'll talk about today, it is very clear from the genetics that the mutation is what's causing the disease. And the mechanism of simply replacing that gene with a working gene is going really at the heart of the problem. No pun intended. Our goal is to improve heart rhythm, heart output, improve symptoms, exercise capacity, ability to do daily activities of living, and quality of life. Now, these gene therapies are a one-time treatment. They take advantage of a mechanism that is designed to deliver a gene to the nucleus with a long-lasting durability. So there is no need for a re-dosing. So we have the potential for a very long-lasting response. A question we get frequently is, if you get treated, does that mean your children will also have the benefits? And the answer is no. So this gene isn't delivered to the egg and sperm cells that would be passed on to your children. There have been very notable and encouraging successes for AAV-based gene therapies across a variety of types of diseases affecting different organs. I've listed them here. I won't go through the entire list, but it is encouraging the broad base of diseases that can be effectively treated with gene replacement therapies using AAV. There's been extensive clinical experience with over 5,000 patients with treated with AAV-based gene therapies to date. One that we focus on is called Zolgensma. It's for a disease called spinal muscular atrophy. There have been over 3,000 patients treated in 50 different countries in clinical use of Zolgensma. Now, Zolgensma uses a particular type of AAV delivery called AAV9, which is also what we use in our gene therapy products, which are called TN201 and TN401. So we are very encouraged by the successful use of Zolgensma with high levels of clinical efficacy, meaningful differences for patients, and a well-established safety profile. I'll start first talking about clinical results we are getting in our first in-human trial of TN201, a gene replacement therapy for hypertrophic cardiomyopathy in people affected by mutations in the MYBPC3 gene. We had excellent results in the mouse model of this disease, where a single dose would reduce the heart mass. Increased heart mass is a key feature of hypertrophic cardiomyopathy. It restores heart function and improves survival in the mice. That led us to this first-in-human clinical trial called MyPeak-1. In this trial, we dose patients sequentially, individually, one at a time. First 3 at the starting dose, which is dosed at 3 E13 vector genomes per kilogram of body weight. And then we've had a DSMB review, an independent safety panel review the data from that first cohort, and agreed with us that it was appropriate to continue on and go to the higher dose cohort 2, 6 E13 vector genomes per kilogram. And we are actively enrolling the second cohort now, and have treated 2 patients, and the third is scheduled. So today we'll talk about initial data coming from the first cohort. In the second half of the year, we'll be positioned to share initial data from the second cohort, as well as longer term effects in the first cohort. The protocol does allow us to expand one or both dose cohorts, and we'll make that decision after we analyze the preliminary data from the second dose cohort. Now here we're showing the baseline characteristics of the first 3 participants in our trial. We are, of course, indebted to the people who have chosen to participate, hoping for benefits for themselves, but also on a mission to advance science, to potentially spread benefits to similarly affected people throughout the world. All 3 of these people were affected by the MYBPC3 mutation at young ages. They have had a more severe phenotype than the average hypertrophic cardiomyopathy patient, with young onset, with high risk of arrhythmia, so that all 3 have required implantable cardiac defibrillators. They all have required open heart surgery. So this is a disease where the excess heart muscle can actually obstruct the flow as it tries to go, the blood goes through the heart. And they had a procedure to remove that obstruction, that extra heart muscle. The surgeries worked, the obstruction was removed, but despite that, they remained symptomatic. For example, their New York Heart Association class is elevated, indicating that they have a high burden of symptoms, and their routine activity is limited by disease. They also have elevated levels of circulating biomarkers that indicate continued stress on the heart and a risk of future cardiovascular events. Those biomarkers are NT-proBNP and troponin. We are happy to say that TN-201 has been well tolerated in the first cohort of patients. There have been no significant cardiac safety findings related to TN-201 to date, no signs of heart toxicity. Importantly, no thrombotic microangiopathy, which is a specific type of immune system reaction that can happen with AAV-based gene therapy. And that can be associated with low platelet count, known as thrombocytopenia. So we are fortunate not to have seen either of those issues. The safety findings we have seen associated with TN-201 gene therapy are abnormalities in blood tests measuring liver enzymes. So the immune reaction to the gene therapy does create some liver inflammation, and we can see that in the blood tests. This is very well known and understood for AAV-based gene therapies. It's consistently effectively treated with immunomodulating therapies, in particular steroids, and that has been the case with TN-201. Basically, we treat patients preventatively, prophylactically with immunosuppression, including steroids, before the gene therapy treatment. And as we taper that off, we can see increases in the liver function tests, in which case, when that happens, we pause the taper or give a little extra steroid, and that has been very successful in controlling this inflammatory liver response. In the field and in TN-201, this prevents long-term complications, and we have not seen any long-term liver complications from our trial. We have had 2 SAEs, serious adverse events or serious health events during the trial that were due to causes unrelated to TN-201. And all participants in the MyPeak study have completed every visit to date and remain in the study. Now, in the trial, we do heart biopsies. We take little teeny snippets of heart tissue to determine whether the gene is being delivered and expressed in the heart as expected. We have high levels of delivery, high levels of DNA. I'm not showing that data here for simplicity, but very encouraged by the number of genes that we are getting to the heart cells, more than 2 copies for every nucleus in the heart. This data is showing the first step for expressing the gene in the heart, called messenger RNA or mRNA. And we are detecting expression in all the samples that we have acquired. So at this point, we have 2 samples from patient 1 at week 8 and week 52, 2 samples from patient 2 at those same time points, and a single sample from patient 3 at the week 26 time point. So in all cases, we're seeing robust high levels of RNA expression. The assay we are using here is very specific for the TN201 derived messenger RNA. We do not detect the endogenous RNA that the patients have from their one working copy of the gene. The levels that we are achieving here are right on par with other AAV-based gene therapies for cardiomyopathies, in particular one in development for Danon disease. We're also encouraged that the levels increase with time. You can see with patient 1 and patient 2, increases in expression between week 8 and week 52. This could even continue into the 2nd year. We do not know at this point, although we do know from that Danon disease study that many patients have higher expression in the 2nd year and continued stable expression out even 3 years. So this is, I guess I mentioned, a one-time therapy, and everything is pointing in the right direction that the effects could last for many years. All 3 patients in the trial have become less limited by their disease, as reflected by reductions in New York Heart Association class. So all 3 at the latest endpoint, so 1 year or week 52 for patients 1 and 2, 6 months for patient 3, all 3 are New York Heart Association class 1, which means they are not limited in their activities by their disease. I'll point out patient 2 who started at New York Heart Association class 3. That is defined as symptoms with less than ordinary activity. So very limited by the disease. And so these are big jumps between classes, and the jump from 3 to 1 indicates very substantial improvement. So we are very encouraged that these patients are feeling that much better. In terms of a lab test, these are the results of cardiac troponin I. Troponin is something that is released by the heart when it is being injured and strained. And we can see that in 2 of the 3 patients, there have been substantial reductions in troponin. These are all high at baseline due to the disease, hypertrophic cardiomyopathy, and 2 of the 3 patients have had more than a 60% improvement in troponin levels. Patient 2 well within the normal range for the blood test now, and patient 3 very close to the normal range. We do take pictures, use echocardiography to look at the structure and function of the heart. We're highlighting one piece of data from that here, and that is the thickness of the heart muscle. In particular, we're looking at the left ventricle part of the heart and the posterior wall of the left ventricle. This excess thickness is a key feature of hypertrophic cardiomyopathy. And you can see that in patients 1 and patient 2, there's an encouraging trend towards normalization of wall thickness towards the normal range. Patient 3 has only been followed to 26 weeks at this point, so we look forward to more data from patient 3. And overall, when we look at multiple parameters of cardiac hypertrophy, we see improvement or stabilization. So again, we're very encouraged by the imaging results from the gene after the gene therapy. So this is the first ever clinical data for gene therapy for hypertrophic cardiomyopathy. At this dose level, 3E13, we have seen that it's been well tolerated. And as I mentioned, the independent panel called the DSMB endorsed dose increase to 6E13, and that we have now dosed 2 of the 3 patients for that cohort, and look forward to dosing the 3rd patient soon. We see encouraging patterns going in the right directions across biomarkers, measures of cardiac structure and function, and we are very encouraged by the symptomatic improvement that we see with the New York Heart Association class. So we very much look forward to sharing more data in the second half of the year. The first data we'll get from the high dose cohort and longer term data from the low dose cohort. Again, we're saving questions for the end, so please write them down. I'll transition here to our second gene therapy program called TN-401, which is for arrhythmogenic right ventricular cardiomyopathy, in particular patients affected by mutations in the PKP2 gene. I will say that the TN-401 gene therapy is very similar in how it delivers the gene to the heart to the 201 therapy I just discussed. So it uses the same platform, AAV9, and the same manufacturing process. So the encouraging early results we're seeing for 201, we are cautiously optimistic. We'll read through for TN-401 as well. We are actively conducting the trial, but we do not have data to share with you today. We expect that in the second half of the year. But we are very encouraged by the robust effects we have seen in the animal models, similar to the robust effects we saw for the MYBPC3 gene. So in the animal model, they develop a disease very similar to human ARVC, including failure of the ventricle, the function of the right ventricle and the left ventricle, and frequent arrhythmias. And a single dose of the mouse version of TN-401 or MTN-401 prevents the failing and scarring of the ventricles, reduces ventricular arrhythmias, and improves survival. And we show some example EKGs here, a normal healthy heart mouse EKG on the top, the EKG of mice lacking effective PKP2 genes, where you can see frequent premature ventricular contractions or PVCs, and couplets, so pairs of these together, and then runs of non-sustained VTAC called NSVT. But again, a single dose of the mouse version of the gene therapy prevents these arrhythmias, as you can see in the bottom tracing. So we announced the dosing of the first patient in the study in November. As I mentioned up front, we do dose patients sequentially, one at a time. It's called sentinel dosing, and review the effects in each patient before dosing the next one. So we are well underway in that process. Again, we are indebted to the patients who choose to participate in these trials, and they clearly helping the community as well as potentially themselves. We have multiple trial sites in the United States as listed here. We tried to emphasize geographic accessibility. We are opening trial sites in the United Kingdom as well. If you want more information about our trials, you can go to clinicaltrials.gov. And use the NCT number listed here, or you can visit www.arvcstudies.com. The eligibility criteria for this trial, we're only enrolling adults in this first in-human trial. Participants must have a mutation in the PKP2 gene that's classified as either pathogenic or likely pathogenic. And need to be significantly affected by the disease, including having frequent premature ventricular contractions over 500 per day, and must have a functioning ICD, and low levels of AAV9 neutralizing antibodies. So just to explain that, these AAV9 vectors that we use to deliver the gene are present among us in the human population, and a small percentage, our data says about 20–25% of patients have already formed antibodies against AAV9, and those might prevent the effect of the gene therapy, might block the beneficial effects of gene therapy. So we do exclude patients that have higher levels of these antibodies. The key goals are to assess the safety of TN-401, to do a dose ranging, very similar to what I discussed with TN-201, and explore for initial signs of efficacy, both on signs and symptoms of disease. This is clearly a simplified list of the eligibility criteria. An investigator will run through a more detailed list with each participant or each patient interested in participating. So you, family members, or people you support may be considering participating in a gene therapy trial. We encourage you to learn as much as you can about the particular study. So there are frequent study visits in first-in-human studies, including a brief hospitalization at the outset. The hospitalization is purely to allow frequent monitoring of the immune system and making sure everything's under control. The feedback we get is mostly that it's pretty boring. But certainly very important to do that monitoring early. And then there are frequent visits back to the study site after that to monitor, both safety and efficacy. It's important to think what expectations you have about the outcome, what you're hoping for, and also how you feel if the results aren't what you hoped or expected. Very important to discuss all the risks, and the investigator at the trial site will have the most information. We share a large compendium of information about the potential benefits, about the potential risks and the mitigations to avoid any complications of the gene therapy, and they can discuss that with you. Certainly having a good close relationship, good communication and trust in the clinical team at the trial site is essential. So definitely look for that. And then your own support network in your community, in your family, who can help you, give you a perspective on a decision on whether to participate or not. Support network is very happy, very, very helpful. I will say that that Tenaya does support travel to study centers. Obviously, all the treatment are covered, and for folks that are traveling further to a study center, that is covered. Now, there are potential risks to participating in an AAV gene therapy trial. We've talked about the immune system. The immune system thinks this might be an infection and tries to attack it. And so that's why we use prophylactic treatments, immunomodulatory medicines, to limit that effect. But if the immune system does become overactive, it can prevent the gene therapy from working. It can create inflammation, such as in the liver that we discussed. So it is critically important that our participants in the trial go to the study center frequently, have the appropriate laboratory tests to make sure that the immunosuppression is doing what is needed, but to also help get the immunosuppression doses reduced quickly, get that tapered off as quickly as possible. Currently, an individual can only get an AAV-based gene therapy once. When you do get it, the immune system will create those antibodies that I mentioned before, the neutralizing antibodies, and those will then prevent the efficacy of a second dose. There's a lot of work in the field to enable repeat dosing of gene therapies, but it's not available today. The AAV mechanism does deliver genes to different tissues throughout the body. For our AAV-based gene therapies, we use a switch, an on switch called a promoter. That means that the genes we're delivering only turn on inside heart muscle cells. And of course, our gene therapies are still under investigation. Not all the risks or possible risks are known today. For the last section here, before questions and answers, I'll make an effort to describe how gene therapy works, in particular, how AAV-based gene therapies work. Essentially, what we're doing is delivering a new copy of the gene using an AAV packaging system. So proteins from an adeno-associated virus that have been designed by evolution to deliver genes to human tissues. We remove all the viral components from the inside. We replace it with the working gene, in our case, either MYBPC3 or PKP2. This is administered through a single one-time IV infusion, spreads throughout the body, but the AAV9 that we use has a high propensity to find heart muscle cells. And when it gets to a heart muscle cells, it's taken up by the cell, routed to the nucleus, which is where all the genes are located. The protein part, the capsid, is broken down, leaving just the gene, and then the heart's own machinery takes that gene and creates new protein. Now, that's a lot to take in. We've been working on other ways to describe how this is working that could be more tangible. And so this is the metaphor we use about building more chairs for your dining room. So say you don't have enough chairs to go around. You're going to host a big party and you need to build more chairs. In this case, the chairs are the protein that is missing in the cardiomyopathy. So MYBPC3 protein or PKP2 protein. Heart cells have a broken gene. They're not making enough protein. They don't have enough chairs for the heart cells to do the function that they need to do. So what do? Well, you order instructions to make the chairs. Now, those instructions will be delivered in a package. So that package is the same as the AAV9. The AAV9 capsid or protein is basically a box where the gene or the instruction manual is packaged up, and it has a delivery address on it that will deliver it to your house, or by analogy, the heart muscle cell. Next, you get the box at your front door, you open it up, you take out the manual, and you take it into your workshop. So that's the process of heart transduction, endocytosis, where the AAV capsid is brought into the heart cell. It's delivered to the nucleus, with the workshop essentially in the cell, and then the package is discarded, and what is kept is the gene or the manual. In this case, you need to make quite a few chairs. So you've invited your friends. You need to make many copies of the specific instructions for that chair you want to build. So those copies that you actually bring into the workshop and use and read as you're building, that is messenger RNA. So that in the AAV delivers the gene, that form of the gene is in DNA, but then that is copied, and one copy of the gene can be used to make many copies of the messenger RNA, and then the messenger and RNA will be what's used to make the protein. So you bring your instructions to the workshop, you give it to your friends, and everyone assembles chairs. Now, in this, you're assembling chairs from parts that are already available in your house. And in the cell, those are called amino acids. Those are the building blocks for protein. So those come from your diet. They're routed to the heart. They're ready there and available. So when the heart gets the mRNA instructions, it can assemble the amino acids in the correct order to make PKP2 protein or MYBPC3 protein. So the result is an increase in chairs or protein levels, and that can restore normal heart function. So I hope that analogy is helpful. That was the last part of the presentation, and I very much look forward to your questions. In the meantime, please find us at hcmstudies.com, arvcstudies.com. We are on social media as well. And with specific questions, please reach out to patient.advocacy@tenayathera.com.
Thank you very much, Dr. Tingley. Very, very comprehensive overview. I have a couple questions to get us started. Let's see. Catherine asks, what does the time frame look like from stage 1 clinical trials to FDA approval, and how long can it take on average?
Yes. So in the case of developing gene therapies, the FDA has many precedents of allowing more rapid progression through the trial stages for efficiency so that patients can get access to these types of therapy most efficiently. So in common typical drug development, there's phase 1, the first in human study, phase 2, which is what we call proof of concept, and then phase 3, or the pivotal study, which is the one that provides all the information the FDA needs to decide whether to approve the therapy. In many gene therapy programs, that's broken down into 2 stages, so they get the name of phase 1, 2. So the studies I described here are basically combining the first in human safety study with that proof of concept. So phase 1, phase 2, and we anticipate that our next studies for both programs will be pivotal studies, equivalent of phase 3, that will allow a full decision for approval. So, that still takes a few years, but, hopeful that it's not a decade or anything to that far out. So I know people are eagerly waiting. It does take some patience. We do need to make sure we are doing this safely and we're gathering all the appropriate information as we go to make sure that we are maximizing not only safety, but we're getting the dose correct, and that will provide the FDA and clinicians and patients the information they need to make decisions in the future for approval and beyond.
Thank
I
you.
will say that the FDA recognizes the clear needs and values for efficiency here. They definitely also recognize the patient voice, and there are important meetings that the FDA hosts where patient, I know SADS has done these meetings, so thank you for that. And they listen very closely. One thing that they have been using is called accelerated approval. So before there's definitive final proof, but when there is initial strong suggestions that the data indicates that it will be very effective, they will give an accelerated approval, which will allow patients access to the gene therapy while the final large pivotal studies are still being completed. So we very much appreciate partnership with the FDA and the fact that they've listened to patients and really focused on speeding up the process.
Okay, thank you. Another question, will the PKP2 trial have the same number of patients as the first 2 HCM cohorts, 3 and 3?
Yes, it's 3 and 3, but then the possibility of expanding one or both dose cohorts.
Okay. And what does long-term follow-up look like for the PKP2 trial?
Yes, so in both trials, we have a key endpoint, a look at the data that's 1 year after the dose, but the protocol includes 5 years of follow-up. So our participants will be going back over the course of 5 years. I did emphasize that there were fairly frequent visits. That's really just in the first half of the first year has the most visits, and by the end, it's a once-a-year type visit. So it's not as burdensome as it might sound on the face of it. But it's important for us to measure the long-term efficacy of the drug so we understand how durable it is.
Thank you. You mentioned sequential dosing. How long does it take to review results before dosing the next patient?
Yeah, so we gather 4 weeks of data from the first patient and then spend a couple weeks analyzing it, reviewing it, including a review with an independent expert and the investigators. So the typical interval is more like 8 weeks between patients.
Okay, thank you. Is there any way to get rid of AAV antibodies once they are present?
So this is a big focus of the scientific community. And for diseases where tissues have high turnover, like the liver, there can be loss of effect over time. And so it's critically important that we learn how to do this better, to do the redosing, get rid of those neutralizing antibodies. There are a variety of approaches being addressed. Plasmapheresis, it's like dialysis, so it's a filter to filter the blood, to filter them out. There are enzymes that can break down antibodies that can be administered. There's trials looking at pre-treatment before the dose that would prevent the formation of the antibodies. So many different approaches. So I look forward to breakthrough results, but I can't say how long that will take.
Thank you. Are there any lasting effects from heart biopsies?
Heart biopsies are done very routinely. Many folks on the call may have had relatives or friends with heart transplant. This is our best way to detect whether rejection is happening in the heart. And so at the study centers where we are running this trial, they are all transplant centers with a high degree of expertise. They do this routinely. It's an outpatient procedure. There are small risks from the procedure, but again, it's very, very safe in competent hands that have done this frequently. The piece of tissue is measured in millimeters compared to a heart, which is much more fist size. There's no, significant loss of muscle or function from the heart biopsies. Our trials have 3, so quite few compared to a heart transplant patient. So and we've had certainly no complications from the biopsies.
Thank you. So one person asked, if I'm not accepted into one of the first phases of the trial, is there a chance that I may qualify for later stages or phases?
Yes, definitely, very much a chance. So the later phases will be larger. They have more patients, so just more opportunities mathematically. And oftentimes we broaden the eligibility criteria. We start our first in human trial in a more focused group where it's, for example, easier to measure efficacy. And then for the pivotal trial, we go to more broad eligibility criteria. But if you aren't eligible for, for example, you have high levels of the neutralizing antibodies, then probably will mean you're not eligible for the pivotal trial as well. So it really depends on the reason that you were ineligible.
Okay, I don't see any other questions at the moment that have been sent to me or posted in the chat. So I will thank you for your time and your fabulous overview for our community. If you have other questions, you can let sads@sads.org know, and we can pass those on to Tanaya. So thank you, Dr. Tingley. We appreciate your time and efforts, and have a good evening.
Thank you, Marcia. And thank you, everyone on the call.